Hey everyone, it's Dr. Marcon with the discussion on Laboratory 24. Lab 24 being about the ECG, electrocardiogram, as well as ECG and pulse. We use the BIOPAC. We've had experience with the BIOPAC to take measurements for an ECG as well as for pulse measurements. So we know that an electrocardiogram provides a view of the electrical conductivity of the heart, or an electrical view of the heart. ECG and EKG are actually synonymous. There are parts of Europe, specifically Germany, that spell the cardio part in electrocardiogram with a K. So you might hear the term EKG being used to describe this method of viewing the electrical conductivity of the heart. It's the same. ECG and EKG are the same. So here we see a typical ECG tracing that we would get once we put the leads onto our patient. On the left, you see a representation of Einthoven's triangle. This is a hypothetical triangle created around the heart when electrodes are placed on both arms and one leg, and the sides of the triangle are numbered to correspond with the three leads or pairs of electrodes used for a recording. So each two electrode pair will constitute one lead with one positive and one negative electrode, and then an ECG is recorded from one lead at a time. So one electrode acts as a positive lead, and the second will act as a negative lead. I'm not going to get into too much detail about Einthoven's triangle. I will talk about that in the discussion or in the lecture. But here we can see the different intervals that make up an ECG tracing. I like to say EKG. Just, I don't know, for me it sounds a lot better. But anyways, you can use either. So we have different segments and waves and deflections that represent the different activities of the heart, and we'll get to that in the next slide. So just to define some of these terms, a wave is a deflection above or below baseline. A segment is the baseline between waves, and an interval is the combination of waves and segments. So we'll start off first. So the x-axis being a representation of time, and then the y-axis is voltage, and that's measured in millivolts, and usually time is measured in milliseconds. So the first wave that we're going to talk about is the P wave. The P wave represents depolarization of the atria in response to an electrical signal coming from the SA node being the pacemaker cell that generates the rhythm and rate of the heart. So first we have depolarization of the atria, and that is shown, and that you can see that visually on the EKG as the P wave. And then the PR interval is the delay of the AV node. Remember, we have the electrical signals that spread throughout the atria, and then they stop at the AV node. So this PR interval represents the delay of the AV node, and this kind of pause allows for filling of the ventricles. Then we have our QRS complex. So Q being that first deflection downward within this complex, and then we have this R peak, and then the next deflection would be S. So this QRS complex representing depolarization of the ventricles, which triggers the main pumping contractions of the ventricles. Then we have a segment, the ST segment here. This is the beginning of ventricular repolarization, and normally it should be flat. And we know that we have conditions that could elevate this segment, such as an ST elevated myocardial infarction. So we see ST elevations in MIs that we can see on EKGs. So the ST segment, again, beginning of ventricular repolarization, and finally we have the T wave representing ventricular repolarization. And then on some EKGs you may or may not see a U wave. We're not going to get into that, but it is possible to see another wave there. But this, for the most part, would be a normal EKG and what these waves and segments represent. This lab, we're going to be measuring EKGs and pulse using our BIOPAC. Had we met in lab, we'd get out those huge BIOPACs and attach the electrodes and have it all set up. The first thing we're going to do, we would place a sensor on the right hand. So here we see the sensor. It's called a plethysmograph. The plethysmograph going on the right hand. We would then put electrodes on the wrist of the right arm. It would be a white electrode, white representing a negative. And then we would put a positive electrode on the left ankle. This would be a red electrode representing the positive electrode. And then on the right ankle, we have an electrode there, and that would be black, and that is our ground. Okay, so we place the electrodes on the volunteer, attach electrodes as well as the pressure monitor. We would then, of course, calibrate our BIOPACs and then begin an experiment. So the experiment involves having a volunteer sitting with their hand on the table about the level of the heart. And then we would measure for 10 seconds. The second situation involves dropping the volunteer dropping their arm below the waist, waiting for about 1 minute, and then measuring for 10 seconds again. Next condition, we would have the volunteer hold their hand above their head for about a minute, and then measuring again for 10 seconds. We would then use the measurements and then fill out the tables, the data tables, to analyze the data. We would fill out the RR intervals, the heart rate, the pulse interval, and the pulse rate. So here are the different conditions that we would have the volunteer going through: them sitting at the table with their hand on the table about the level of the heart, and then having them sitting with their arm below their waist, and then sitting with their hand above their head. So this is what we would expect to see with our BIOPACs. The top being the EKG and the bottom showing the pulse peaks. Okay, so we would want to measure between, for the EKG, for example, we would want to measure the R, this R peak right here to R peak, and we would highlight that, and that would represent the RR interval. Okay, and then the next we would want to highlight the area between two successive pulse peaks. So here we see a pulse peak, and here's the next pulse peak, so we would highlight that area there. So we would get the actual heart rate when we measure the RR interval, and the computer could calculate the heart rate when we highlight this area here, and then we would get the pulse rate when we highlight the area between the two pulse peaks here, and they should be about the same. What's great is you can actually see that this QRS complex represents ventricular contraction. So this is the force that is pushing the blood out of the heart, and the pulse represents that that pressure that's finally arriving at the tip of the finger here. So we know that once the ventricles contract, that pressure will kind of take a while to arrive at the fingertip, but as we can see, this ventricular contraction generating the pulse eventually. Okay, so that's actually kind of cool. I think everything's cool. So we are able to measure the RR interval. That'll give us the heart rate, and then we measure the pulse interval, and that will give us the pulse rate. So we would fill out the table with our data, and that will help us. We fill out the data for the 3 different conditions that we talked about, and using this data we can then fill out the information and look at various aspects of the data. This next slide, so we want to just concern ourselves with the box in red. So we want to compare the typical heart rate measured under various conditions, as well as measurement of the systolic peak. That's the systolic pressure, measuring the peak of R and of T, as well as diastolic time, which is the T to the peak of R in seconds during sitting, at rest, and after exercise. So this next part, we want to take a look at systolic time and compare it to diastolic time. If we look at the instructions here, we can see that this next part, we want our volunteer still hooked up to the EKG. We have them sitting with their hand on their table and then measure for 10 seconds. And then the next condition, we want the volunteer to take 3 deep, slow breaths, mark the beginning of inhalation with the F4 key, and then exhalation with the F5 key. We then want to unhook the electrodes and have them exercise for a bit, and then have the volunteer be reattached to the electrodes and then measure the EKG for 60 seconds. We fill out the data tables 1 through 3. Table 1 is beats per minute. Table 2 is systolic and diastolic time. Normally we'd have time permitting in the lab to kind of fill out this table for the EKG components, but for your lab quiz, we're just going to be concerned with the box in red, the 2 boxes in red. So when we talk about systolic time and diastolic time, we look at the different intervals that represent either ventricular systole and ventricular diastole. So the EKG estimates duration of systole and diastole of the heart. So we're looking first at the QT interval. So the QT interval represents ventricular systolic time. And here we see that we can measure the time. It starts at about 100 milliseconds, and then at the end of the T wave, we see that it ends about 350 milliseconds. So when we calculate that, we just get the difference between those 2 numbers. So in this case, the QT interval equals 250 milliseconds, or 0.25 seconds, however you want to record it. But 250 milliseconds, also the same as 0.25 seconds. To get the ventricular diastolic time, we take the TQ interval. So the TQ interval begins at the end of the T wave here, and we can see it's about 350 milliseconds, going all the way to the peak of the QRS complex. So this representing the ventricular diastolic time. So the TQ interval starts at about 350 milliseconds and goes to about 900 milliseconds. We get the difference of that, and that's about 550 milliseconds. So just remembering our formula for mean arterial pressure, we can see that mean arterial pressure we know is 1/3 systolic pressure plus 2/3 diastolic pressure, which makes sense because the majority of the time the heart is in diastole. This is because the heart is taking its time to fill with blood. So 2/3 of the time the heart will be in diastole, so filling with blood. 1/3 of the time it will be in systole or systole, however you like to pronounce it. Okay, so again, EKG estimates duration of systole and diastole. We are able to measure the time in each interval based on the conditions that we have our volunteer going through, and then we place those times in the table. And that's the discussion, or that's the data, or data collecting for Lab 24, and we'll go into the discussion in just a bit.